So we're going to get going. Thank you all for joining us today. This is the first of our three-part series, webinar series on neonicotinoids, a pesticide that, as Dr. Bredeson will review, is one of the most abundantly used pesticides in the U.S. And he is going to be, I'm not going to steal anything from him. He's going to give us a lot of his research he's done through his graduate school career and on into his professional career. Currently, he is a faculty member, a professor at St. Cloud State University, where he teaches ecology and conservation science to the students there in St. Cloud, Minnesota. And we are grateful to have him here today to share with us his knowledge of neonicotinoids. So without further ado, I will turn it over to Dr. Bredeson.
Okay, Julio, Ryan, can you see me and hear me okay?
Yep, and we see your slides just fine.
Okay, awesome. Well, thanks so much for the introduction. Thanks for inviting me. For everyone that's speaking online, I'm also standing in front of a room full of people. Okay, so there's eyeballs staring at me. So I'm gonna try and do things tactfully, right? And make sure and do right by both audiences here. But I'm not gonna walk away. I'm hopefully gonna stay loud and clear for all of you. And if I do say like ask a question, which I like to do to keep everyone engaged or to have a show of hands, please, everyone that's online, like you have that chat bar open. Feel free to write stuff in there. Ryan's going to be taking note of questions of people if you want to use the little hand emojis and stuff like that. If I have to ask for a show of hands, so let's try. Even though we're remote, let's try and be as interactive with this as we possibly can. So today, this group of people, Ryan, okay, asked me to come and talk about something that I've been thinking about and looking at for a long time now. It's amazing how time flies. Something called neonicotinoids. Now, show of hands in the room, and if you're standing in front of your computer at home, show of hands too. How many people have heard of neonicotinoids before? Okay, in my captive audience in the room, it's 100%. People have heard of neonicotinoids. So we'll see. Maybe you guys will already know everything I'm going to say. So maybe this will be boring. I'll try and be animated and make something new for you. What are we going to learn today? Well, first of all, and maybe most importantly, right off the bat, I think Ryan told me, we want some nuts and bolts information on what is a neonicotinoid. Okay, where did they come from? How did they work? Where did they go? All these kind of nuts and bolts about this thing that we hear about so often. We all hear the term neonicotinoids, or maybe more commonly neonics, but let's maybe dig in to the nitty gritty, some of the fundamentals about what a neonicotinoid is. I kind of like the nitty gritty of stuff, and then we can build up from there, right? We're gonna really with a laser focus, and then we're gonna keep doing this step back. Okay, but we're going to start with the laser focus. We're going to then move on to a story about neonicotinoids in a situation that they were designed to be in an agricultural field. Okay, how do neonicotinoids behave when they're in an ag field, in a situation interacting with crop plants and the insect community that exists in that crop field? Then we're going to start to get a little sneaky, right? And start to understand and figure out, okay, we know how neonicotinoids are supposed to behave. What happens when they start to do things that are kind of curious? We find them in places that we weren't sure they were supposed to be, right? And I'm going to tell that story with a cover crop talk, okay? That's one of my favorite studies I've ever done. And then we're going to get into the thing that I'm into as of late, okay? We're going to talk about in-field neonics, movement of neonics, and all of a sudden we're going to start to talk about, okay, there's a lot of information coming out on neonicotinoids ending up in places that we never would have dreamed them being in. Okay, some of that work, importantly, even coming out of this lab that I'm standing in right now. We're going to talk about, okay, how, that's a head scratcher. How on earth in deer, in predators, okay, we're starting to measure these compounds. How do they get there? Okay, I'm going to start to sort of hopefully draw a line from point to point and bridge our gap of knowledge with that last story. Okay. As our little bee fly friend says here, please ask questions throughout. Okay, don't be afraid to raise a hand. Don't be afraid to interrupt Ryan. Okay, he's going to be moderating and he'll jump in if people have questions. Okay, I'm already finding myself running my mouth a little bit, and I've been told that you guys, it's 1 o'clock here where I am, and you guys are supposed to leave work at 4 o'clock. So that's okay. I should be done about by then. Okay. So. All right. So what are these things? Neonicotinoids. I may refer to them as that. I may refer to them as neonics. Well, they first hit the scene in the early 90s. They're developed by the Bayer Crop Science Company. Okay. And they are a class of insecticides. Okay. I like to think about neonicotinoids kind of like a box of chocolates. Okay. So a box of chocolates has a bunch of sweets in it, right? But there's a few that aren't exactly the same. And some of those sweets, right? The orange creams, some people like those and they work really good at satisfying your sweet tooth. Others, you know, maybe there's a peanut cluster that gets me for sure. Okay, so all of these things are sweets and they take care of your sweetness craving, but they act differently for different people. Well, neonicotinoids are very similar. Neonics are like a box of chocolates, and there's a bunch of different kinds of neonicotinoids that are more useful, more let's say, adapted to certain situations. Okay, so I'm from Minnesota. In Minnesota, we have 5 registered neonicotinoid active ingredients. And neonicotinoids are incredibly versatile. Okay, we'll talk about that more on the next slide. But we use these things. You can put neonics in soil drenches. You can inject them into trees. You can put them as powder granules in the soil, and you can even so there's tons of them. There's over 500 registered products in Minnesota that contain a neonicotinoid pesticide. Okay, but the big 3, okay, are seed treatments. This is by far the most common use of neonicotinoid active ingredients. How they enter an environment is through a coating on the outside of a plant seed, and that seed is placed into the ground. These are the top 3: imidacloprid, thiamethoxam, and clothianidin. And you'll hear all 3 of those during the rest of the conversation today. But just so you know, right, those are just individual chocolates, part of the chocolate box that is neonicotinoid pesticides. Okay, and it's really this one here that has kind of taken the world by storm. The neonicotinoid imidacloprid. Its trade name on a seed treatment is called Gaucho. There's thiamethoxam, clothianidin. You can see their trade names there. I used to tell people, especially a lot of farmers, you know, make sure you're aware of what's on the seed that you're purchasing, right? Because it's not, you'd think it'd be really easy to tell. It's not that easy to tell. Okay? And it's only getting a little bit harder to tell what is actually on the seed that you purchase, because they've even sort of gone away from utilizing some of the trade names and gone with more trade names that correspond to a medley of pesticides on a given seed. Okay, so there can be a suite of different fungicides and insecticides, and they'll have a new trade name associated with it. So you really do your homework on that. Okay, so seed treatments are this method of pesticide application. What happens when you put a neonicotinoid pesticide and you coat it to a seed? I've got this jar in front of me. These are sunflower seeds and they're colored blue. Okay, that tells me, that indicates that there's something else besides just sunflowers in this jar. Well, there's a neonicotinoid seed treatment on those sunflower seeds. Okay, and when you plant a treated seed into the ground, that neonicotinoid is highly water soluble. Think about a sugar lump in your water, okay, in your hot water, or salt in soup. It dissolves, right? Well, the same thing happens when you drop a neonicotinoid coated seed in the soil. Rain comes along, or soil water percolates up, and that neonicotinoid then becomes part of the water solution, okay, that hydrologic environment. And that is how the neonicotinoid enters a developing plant, is through the roots. They dissolved in soil water. Okay, the plant is sucking up that water that's got a local high concentration of a neonicotinoid. Okay, so as you can see in this little cartoon on the bottom here, there's a treated seed. You plant it, it grows, and then throughout its existence of this plant, the aim is for that neonicotinoid pesticide to become more dilute per unit tissue throughout the life of that crop plant. That's the aim, okay? And the aim is also to prevent exposure between the pesticide and beneficial insects. The aim is to target those insects that are only consuming the plant's tissue, okay? And to avoid those organisms that are not consuming your plant. That's the aim. Okay, so I don't like this slide, so I'll try and make it sound better than it looks. Why do we utilize seed treatments in the first place? There's other methods of insect control, aren't there? Okay, well, first and foremost, we utilize seed treatments, or they are used because, well, some insect herbivores are subterranean, they're below the soil. Okay, or maybe like this European corn borer, it's protected in some way. It's on the inside of a corn plant, so a spray can't get them. Okay, for other insects, okay, those tissue-consuming insects, we can use genetic tools, okay, because those insects are consuming the tissue, right? And in a genetically modified organism, that tissue actually becomes the pesticide that kills an insect. Okay. But there are this suite of insects out there, right, that don't actually consume, per se, the proteins of the tissues of a plant. They're tapped in with their needle-like mouth parts into a leaf, into a stem, and they're sucking out the water being brought up from the roots. They're sucking out the sugars that are being created by the plant leaves. And they're not actually very well targeted by some of these other types of control measures like BT crops. So in a nutshell, what are neonicotinoids targeting? It's those insects that are below the soil surface and within crop tissue, or not directly consuming proteins from the plant. They're just tapped in consuming the sugars of the plant. That's what neonicotinoids are intended to target. But we have this issue, okay, with an insecticidal seed treatment. It's jumping around a little bit there. That's okay. That there's a flaw, right? Pest management in modern ecological or regenerative pest management, the aim is to develop a system that is robust and diverse enough to prevent insect outbreaks in the first place. Right? Well, the major issue with the utilization of an insecticidal seed treatment is you're using it before the growing season ever starts. Okay? And I was walking out in this pea field that you can see in the video, and scattered everywhere are these pink pea seeds. Well, they're coated with pesticide. And this video is taken, mid-April, late April. There's no plant growth in the field. There's no insect activity whatsoever. But you can see this incredible amount of pesticide already being used in that field before we ever have knowledge of a insect population. We don't know if there's going to be an issue at all. Okay, so that's the first aspect why they're problematic. And then the second is that consistent year after year use of a neonicotinoid pesticide can lead to accumulation of the toxin in the soil over time. Okay. All right, neonicotinoids. They are the #1 pesticide in the world. Okay. By weight, there's more active ingredient of neonicotinoids than any other insecticide produced. There was this great paper a number of years ago from Douglas and Tooker here, and they went through a lot of work to figure out how much, what is the poundage, or kilograms in this case, of neonicotinoids applied to U.S. agricultural fields every year. Okay, and you can see it really, it started in the early 90s and has really spiked up since then. And you can see the main culprits are soybeans, where just under half of all soybean seeds are treated with a neonic seed treatment. And really crazily, in my opinion, almost 100%, almost every seed of corn that goes into fields in the United States has a neonic seed treatment on it. That's pretty wild, okay, because we grow a lot of corn. So let's try and contextualize this a little bit. So what does it really mean that almost every corn seed in the country is planted with the neonicotinoid insecticide? This is what I'm going to have. This is my greatest challenge of the day, is trying to contextualize what this really means. Okay, so to do that, I'm gonna tell you a little bit of a story about corn. Okay, and a specific corn seed treatment called Cruiser Max. And in Cruiser Max, the main ingredient is one of our neonicotinoid insecticides, thimethoxam. Okay, and on every kernel of corn, we have 1.25 milligrams of active ingredient thimethoxam per seed. Okay, and I looked it up. It's like, okay, how much? My job here is to help you visualize. So I looked it up. How much is like 1.25 milligrams? It's like 24 individual table salt grains, about that much. Okay, and that's the amount of the weight of neonic thimethoxam per corn seed. Okay, in this specific seed treatment. Let's do a little bit of math. We've got 1.25 milligrams thimethoxam per kernel. We've got almost 100,000 acres, sorry, almost 100 million acres of corn grown in the US. And then on the low end of the average corn population per acre is around 32,000 seeds per acre. Okay, so if we figure this out, we get a rough, very rough estimate of about how much of this one neonicotinoid type is entering United States soils every year. Okay, it's a lot. I'm not even gonna say it's that many milligrams. There's a lot of zeros there. It's a ton. So let's make that a little bit easier to understand. Okay, it's a lot of pounds. It's a lot of millions of pounds. Almost 10 million pounds of just this single neonic pesticide. Okay, so again, it sounds like a lot, but it's really hard for me, thinking about a person in your situation, to look up at that. How much is? I can't visualize weights very well. Let's look at it in a different respect. So the Minnesota Department of Agriculture has this really cool table, and I've got the link to it at the bottom of this page if you want to look at it. It's got all sorts of pesticides that are approved in Minnesota, and then it tells you what's called the LD50 for a honeybee for all these different kinds of pesticides approved in Minnesota. LD50, does anyone know what LD50 is? Okay, got a couple of people in here. LD, lethal dose 50. So essentially what this means is that when you feed a certain concentration of whatever compound to, let's say, 100 honeybees in this case, at this concentration, half of them will die. Okay, it's kind of a weird toxicology term because there's a lot of variation within populations, and to kill 100% of a population, it's just a lot different than if you were to kind of take the amount that kills about half. It's just a really useful metric in toxicology. So there you have it. I don't know to say. You can all see it, right? That is a very, very small amount of thimethoxam per individual honeybee is what it takes to kill half of a population of bees that consumes that dosage. Okay? again, still a little bit out there, still a little bit nebulous concept to try and imagine. So let's instead take one single corn seed. Okay, one single corn seed, we're able to feed almost 500,000 bees with the LD50 dosage of thimethoxam. And assuming that that dosage will kill half of the bees that consume that amount, then one corn seed will wipe out about 250,000 bees. One corn seed about that big. Okay. So I don't know. Hopefully some of these numbers are sinking in. So the relative toxicity that neonicotinoids can express. All right. I told you that we were going to get into the nitty gritty, and I meant it. I like understanding how something works. Okay, Ryan, can you hear me? My microphone died, so.
Go to
Ryan.
go to your mic. Like
It does
All right,
say.
I was muted. We can hear you now.
Okay, good. All right, I've had to change my.
Reshare your slides.
Yep. Okay, sounds good. Gosh, I hope I didn't bore the speaker to death.
While we got you on a break here, while you're getting your slides, we had one question come in, Mike. They were wondering if you could explain the name neonicotinoids. Where did it come from? And yeah.
Yes, that's a fantastic question, and that will partially be answered on this slide right here. Okay, so hopefully that question will be addressed right now. Okay. So how do neonicotinoids work? I like to remind people, because we get really desensitized, during any given growing season, just the sheer amount of pesticide that we encounter, right? We sort of assume that it's relatively safe, or, oh, geez, a pesticide kills an insect and that's that. Okay. No, I kind of like to make sure that people understand the process. How does a neonicotinoid kill an insect? Okay. Well, let's look at my little cartoon here. And this cartoon took me, I think, an entire day to make. So please enjoy it. Right? So here we have an insect brain, or a bird brain, or my brain, your brain, okay, a fish brain. And that brain sends a message to my finger to do this, right? To the muscles in my forearm to flex and extend. Okay. And that's how this happens. Okay. Please look at the screen. A message, an electrical impulse is sent down a nerve across this space called the synaptic cleft. Sounds pretty cool. And then that message is taken on by another nerve and another nerve and another nerve, and it eventually excites whatever muscle, gland, whatever it needs to happen. Let's say a hormone needs to be released. Brain sends this electrical impulse to the gland, and you get some hormone excreted. Okay. An insect, it needs to escape danger. Okay. It sends an electrical impulse from its central nervous system down to the leg muscles, and that leg muscle moves. It will run. Okay. And it does that. Let's watch it again, because I worked so hard on it. It does that. It bridges this gap. It bridges this gap. Wish I could see that. Anyways, this big gap in the center of your screen, it does that. It bridges that using a neurotransmitter. A neurotransmitter are these little compounds called acetylcholine. That's a neurotransmitter. It's kind of like when you're playing telephone, or you've got one can here and I've got a string, another can to talk to somebody else. It's the string. It's what does the communication from one cell to the next. They cross this gap and they bind to the nicotinic acetylcholine receptors. That's where nicotine gets its name. That's where nicotine from tobacco plants has its activity, is at these receptors. And this is the site that neonicotinoids also affect an organism. Thus the name neo or new nicotinoid. Neo, new, nicotinoid, nicotine-like. So it's something that's new that acts like nicotine. Okay, that's where neonicotinoid comes from. Okay. What's cool about this amazing piece of our physiology is that I'm able to flick my finger and do it again right away, right? Those acetylcholine molecules are taken away and ready for the next impulse almost instantly. It's amazing. But what do you think happens when neonicotinoid enters the picture and is in this unregulated amount in this highly regulated space? Okay, well, they come and bind to those same post-synaptic surface receptors. And instead of being removed really fast for the next impulse, for that insect to run away or for that gland to squirt some more juice again, instead, they stay there. And they lead that next nerve to continually fire, to continually innervate. Okay? This is not good for an insect who needs to have very good control of its body if it's going to, say, evade predation, hunt for prey, move out of the baking sunlight, go get water, things like that. It has to have its wits about it. It has to have perfect nerve function. When its nerves are continually firing, it causes paralysis. It's imagining every muscle in your body contracting at once. That's what happens. Okay, so really what occurs in most, what's postulated, what kills most insects that are intoxicated with a neonicotinoid is actually their physiology becomes starved of resources. Okay, so they essentially starve or are consumed by something else, or they dry out, they freeze. The elements get them in some way, shape, or form because they just can't react with their nervous system in a way that they would normally have to do. Okay? Cool. All right. So where have neonics been found in the environment? We know where they're supposed to be. They're supposed to be in treated crop plants. And yes, they certainly are found there. They're also found in nearby treated plants. This lab has done plenty of that research. We can find them in the dust coming off of planters, in the dust blowing off of fields. We can find them in the soil, lakes, rivers, wetlands, well water, and very interestingly, and Ryan, our moderator, is from Iowa. I always poke him about this. This was one of the craziest stories I ever read, journal articles I ever read when it first came out. Campus water fountain drinking water at the University of Iowa. Okay, the most common neonicotinoid insecticidal seed treatments in the drinking water on campus. Suffice it to say, wherever we look, chances are you can find it. Okay, wherever water goes, wherever soil that's coming off of a treated field goes, you find it. Okay, I'm going to skip this. All right, unless people have any burning questions right now. Question in the room. Go ahead. Yeah, that's a good question. The question is, how long do they persist in the environment, and how long? I had a slide and I deleted it because my talk was going to be too long. Yes. So neonicotinoids, right? They flow in water. Okay. They are wherever water is. There's a few things that degrade them. UV radiation, sunlight can break them up. Microbes will munch them because they're a nitrogenous compound. A lot of microbes look at them and say, nitrogen, baby, it's fertilizer for me. And they eat them, they break them up and detoxify them. So those are the things that break them down. Okay. Radiation and microbial digestion. Things that make them persist longer in an environment is colder temperatures, drier environments. more clay in your soil. Clay has this amazing power to act like a magnet and glom compounds onto itself and keep them there for a long time. So the more clay you have in your soil, the drier it is, you're going to have a longer persistence of a neonicotinoid pesticide in the soil. Interestingly, the more organic matter in your soil, the more organismal life in terms of microbial communities, it breaks it down faster. Okay, but nonetheless, the half-lives in the environment can reach over a year. So if you're doing it year after year, you will most likely see accumulation over time.
Mike,
Okay.
we had a question online real quick
Okay.
to clear something up. I think this gentleman was wondering about risk of honeybees consuming. I think you're going to touch on this, but the question was maybe they didn't, or yeah, exposure. How do honeybees get exposed to neonicotinoids?
Sure.
Is
That's a
it
great
direct consumption of seeds or some other way?
Sure. No, not direct consumption of seeds. We'll talk about a few different pathways. But neonicotinoids are found in a treated plant. It's found in the pollen and the nectar, or it can. Okay. So that's really one of the most direct routes of exposure for honeybees to be exposed to neonicotinoid pesticides. At the end of my talk, I think bring up a really important exposure route that hasn't been explored yet, that I think we're going to find is really important. But I'm not gonna steal my own thunder yet. But yes, that's probably the most important exposure routes are via nectar and pollen. And springtime activities, planter dusts can be deposited all over the place. Lots of honeybee hives are found in heavily agrarian areas. So there's plenty of ways for them to be exposed from through different avenues. But direct consumption, yeah, definitely through pollen and nectar. Okay, so I got to move on here, guys. Otherwise, we will be here till 4 o'clock. I want to talk about this first research study that I had with neonicotinoid pesticides. Okay. I wanted to know, are these things working? How do they react in a crop that is highly entomophilous, which is a term that means insect loving? And sunflowers are exactly that. They're one of our only crops that are derived from a plant or a native plant that's from here. Okay, so there's tons of insect activity that occurs on sunflowers. So what did we do in this study? Okay, it took me 2 years, but I had 3, what we call them, site years. I wish I could do a pointer or something like that here. Maybe I can. Let me try and get my pointer up. Is that seen there? Oh, kind of. Okay, that'll suffice. All right, so 3 years, 2013, 1 group of sites, 2014, 2 sites. And I had half of the sites were treated with the cruiser, with thiamethoxam seed treatment, and half were naked seeds. Okay, nothing on them whatsoever. Throughout the growing season, we'd go out there, we'd grab plant tissue, we'd bring it back to the lab, mush it up, and then analyze, see how much insecticide was in sunflower plants throughout the growing season. We collected tons of insects. We looked at thousands of individual sunflower plants. There's a lot of nooks and crannies on sunflower plants. You could see Claire and Janet up here. I mean, I thought it was amazing. Claire was allergic to sunflowers and really hated this project, but she kept coming out there. And I appreciate that. So we collected so many insects from both types of plots, both treatments. And at the very end of the season, I wanted to know, okay, so farmers are actually spending money on these seed treatments. Does it have a return? Okay, that's kind of the most basic question a person can ask about any given agricultural practice. Is it pay? Okay, so that we're going to answer that question. Okay, so first, let's talk about the pesticide. The answer is Yes. Okay. We look in sunflower tissue. You see it in the tissue throughout the growing season up until about this stage. And remember, thiamethoxam is the neonicotinoid that we actually treated onto the sunflower seeds. So it's there, and I measured it up until plants looked about like this, right at the start of the reproductive stage. Now, to make the story a little more confusing, I need you to perk your ears up right now. Thiamethoxam, right, is the neonicotinoid that we treated the seeds with. Very crazily, very interestingly, in plants, neonicotinoids can be metabolized or slightly altered into other types of neonicotinoids called toxic metabolites. So, excuse me, even though we didn't treat sunflower plants with clothianidin, we see it throughout the growing season. And what's extra interesting is that I've got this red arrow pointing to this sunflower here. I was able to measure the toxic neonic metabolite, clothianidin, well into pollination and seed set. A lot longer into the field season, the growing season, compared to the actual seed treatment that was placed onto the seeds themselves. So that was really fascinating. That was a super interesting finding throughout this study. Okay. Let's talk about insects now. Okay. There's this slide is way too small. So just listen for a second, right? I wanted to look and see, okay, we're utilizing this seed treatment to prevent herbivory, to prevent insects consuming my sunflower plants. Okay. And as a whole, all insects, all of them pooled together, we tended to see less of them in fields that were treated with the neonic pesticide. When we look at these characters right here, which are the main pests of sunflowers, those that farmers should be scouting for and looking because they have potential to cause an economic damage, I didn't see any sort of significant decrease in any of their populations when utilizing the seed treatment. Okay. So now that we know that overall the seed treatment had kind of this effect of depressing the total population of insects in sunflowers, it's perhaps not that difficult to comprehend that when we look at just predators, these are just insects that are out there eating other insects. These are the quote, the good guys that we want to attract to be there as an army to take care of our pest insects. We look at the 3 different field sites, you see sort of this, again, moderating or depressing effect of the seed treatment represented in the white bars. Okay, a little bit of a lowering of the predator population across all the field sites. That's pretty, I guess I could probably expect that. If all insects are a little bit lower when we use treated seeds, I'm going to be recruiting fewer predators to eat fewer insects. Okay, that's kind of, it's intuitive. It shouldn't blow anyone's mind. Okay, this is something though that has sort of baffled me. When we look at the pollinators, okay, the bees, both honeybees, managed bees, lumped together with native bees in the system, we see a similar drop, okay, in our treated fields. We just didn't find as many pollinators visiting sunflower heads. So that sort of reduction made sense to me when we were talking about predators, because there's maybe fewer other insects for them to eat. But here in this case, I mean, pollinators are just visiting sunflowers and leaving. They're taking pollen and nectar and leaving. Why would there have been fewer pollinators visiting treated fields? And this bothered me. This bothered me for quite a long time because, as a researcher, when you set up this study, especially as a student, you're like, did I do everything I could to make sure to remove bias? Was I picking plants that had Was I not seeing pollinators as much? It was hard for me to understand this. Okay. And this did bother me for a long time. I even gave my defense on this project without really having a good explanation for this until I started to dig years later into more research that was coming out and started to see that other people were identifying the same thing, this actual avoidance ability of pollinators to treated plants, which was kind of wild to me. And I guess it shouldn't come as too much of a surprise. I mean, they use bees to sniff out bombs and stuff like that, so they can smell. I mean, their sensitivity is so great. So, anyways, people are researching this, right? They're looking, they're around, they're finding that pollinators are actually perceiving, to a certain degree, neonicotinoids in treated plants and avoiding them. Now, this is a good thing in one respect. Why? Well, why is that a good thing? Right. If pollinators have the ability to perceive an intoxicated plant and avoid it, well, that's good on them, right? They can avoid further exposure to those pesticides. Okay, on the flip side, okay, it could also be bad. Like, in what sense is this maybe bad? Maybe it's not a fair question, but I'll see if anyone wants to take a stab at it. Okay, I think it's bad. Yeah, Will? Right. Well, that's number one. Like, that's not like, pollinator communities are already pretty food limited. Okay, and to all of a sudden make more food be off limits to them is not such a good thing for either managed or native pollinators. The other thing is that plants require animal pollination. The majority of plants of dicots require animal pollination. Okay, so what might this effect have on plants, on productivity of plants? Well, somebody looked into that very thing, okay, in sunflowers, utilizing the same exact seed treatment as I was looking at. Okay, they went out there and they said, okay, in plants that have an insecticidal seed treatment, is it affecting how big the sunflower heads grow? And here in this study, yes, you see a negative effect of a thiamethoxam seed treatment on the size of a sunflower's head. Okay, you look at the overall seed set of that plant that has been treated with an insecticidal seed treatment, it's fewer. So this is really kind of amazing pieces of a puzzle being put together that I don't think anyone initially expected, right, from the pollinator community, and a knock-on effect when it comes to potential agricultural productivity. Okay, and this segues into what I actually found in the study myself too. At the time, I couldn't really explain it. I just thought, well, there's some variability there. Seed treatment didn't make much of a difference, but I always had this nagging thing in my head, like, why would I see in my seed treated fields a little bit less yield? And now I think that we're starting to come up with a pretty good idea of why in these agricultural settings, in settings where the neonics are behaving in a way where they're supposed to, we're seeing some unique side effects. All right, let's stop talking about sunflowers and on to the next thing. The next project, I was kind of done with pesticides. Okay, what I learned from that first study at the time was, hey, you want to use a pesticide, it kills insects. That wasn't very much of a brain buster. When you use them, you reduce your insects. Okay, and if you want to do that, then you've got the tools to do it. That's not hard to do. What I wanted to do, and what I thought was more of a fun challenging project was to attract beneficials, attract the army, okay? Take an environment that looks something like this, something that as an ecology professor I would call a ecological desert, and give it some function, okay? Make it a place where organisms have resources to persist in order to develop communities and the complexity that we see in natural ecosystems that prevent pest populations from going haywire in the first place. Okay, we did a good job of that. We did an amazing job interseeding cover crops. But again, it's amazing how history kind of repeats itself. You get this nagging feeling in your head. All right, I had these wonderful resources for beneficial insects planted up and down corn rows, and I was attracting insects like crazy. But the big challenge, one of the biggest challenges to conduct this research was finding a field where there wasn't an insecticidal seed treatment. It was the hardest part of the entire project. Okay, so why am I studying this system, trying to make more complexity in this cornfield and attract beneficial insects and create more functionality if I know that almost 100% of the fields out there have these neonicotinoid seed treatments? Okay, right? They're almost ubiquitous in corn. And then new research came out measuring how much of the neonicotinoid put onto a corn seed actually made it into that plant. Not much. Okay, 2 to 20% of an insecticidal seed treatment that's put onto the seed makes it into the plant. Okay, I'm bad at quick math, so I just did it beforehand. 80 to 98% of that insecticidal seed treatment doesn't go into the plant. You can't look at numbers like that and not look into it. Okay, you can't look in that if you're a graduate student of good conscience. You just can't let that opportunity pass up. Okay, so it's a simple study, right? It's not hard to find fields that have seed treatments on corn. So what I did is I went out there, I planted this freebie cover crop mixture that I got right smack dab in the middle of treated corn rows. I gave it the best chance it could. The field was treated with the lowest dose of seed treatment, so it had a long way to go. Okay, I had these 6 plots. I went out there when the corn was pretty young and planted these seeds and let them come up. Okay, throughout the growing season, I went out there 7 times and collected 2 of the cover crop species, hairy vetch and cereal rye. And I grew all of my control samples, my negative controls, in the greenhouse. Okay, and maybe this is one of those aha moments I had too. I had to grow my negative control tissue in the greenhouse because I had just had a conversation at an entomological meeting with somebody else that couldn't find neonic-free tissue in the field. Okay, they said, can't be done. Like, all right, fine, I'll grow it in the greenhouse. And that was a big aha moment for me. Okay, so we take that tissue, we bring it back to the lab, we mush it up after lunch when we got lots of energy. Okay, we make this little mush ball into it, we spin it around really fast, and we just take the liquid off the top of it. And we do what's called an ELISA analysis, where we pipette our sample into these wells with a bunch of other chemicals, and it makes the well change color. And how much a well changes color corresponds to how much pesticide is in a sample. And then you measure that color change. Okay, it's kind of a sweet deal. So let's look at the results here for how much, for whether we found pesticides in our interseeded cover crop. Okay. Can we answer our question? Is the pesticide moving from where we put it into something where we don't want it to go? Okay, and yes, okay, this is not going to surprise anybody. This is the compound that was treated on the corn seeds. This is its breakdown product. What's interesting here is in both, you'll find this in both the cereal rye and the other plant, the hairy vetch, we found a lot more of the breakdown product, the toxic clothianidin breakdown product, than we do the actual compound that we treat to the corn seeds itself. Okay, which is really crazy. So when you're all doing your own neonic study someday, it's just one thing you've got to be aware of, right? You don't always have to go snooping around for the thing that you put out there. You got to be aware of, is it turning into something else? Okay, what do I need to be collecting to get the whole story? Okay, and I bring this up because in both plant species, we see a little bit of an increase at the very last sampling, and I think that's because we had a rainfall event right before that sampling period. Here's hairy vetch. The graphs look almost the same. Okay, just to show that it didn't matter which plant type, right? We had a dicot, we had a legume, and we had a grass, and they were both behaving very similarly. Okay, so what did we learn from this study, right? We just got done with sunflowers where we try and figure out, okay, what's happening? How long is this insecticide persisting in the plants that we're actually treating? And now we're starting to move away, right? And start to impact other organisms that we never intended to impact. Okay? And we're starting to identify, at least at this point in my career, looking at neonic insecticide, this is where I started to first identify different routes of exposure for how might these pesticides that are so ubiquitous get into communities and places that we don't necessarily want them to go. Okay, they don't stay put. They don't stay put. Certainly, some of this 80 to 98% of pesticide entered my interseeded cover crops, but there's still more to go around, right? And that's where we start to see these different stories come up of communities of organisms that are not associated with agricultural activities whatsoever being influenced to a large degree by a pesticide. Okay, we're finding them in aquatic ecosystems. We're finding them in tissues of mammals. How is this happening? And that's where we go into my last little bit of work here. This is kind of the last story that I'll tell you. And it's a really simple story, but it's one that's also something I think is really kind of cool. The work is really cool anyways. Okay, we're getting to be the time of year where we start to see a scene like this quite a bit, right? We see in agricultural fields, we see this pools of water. I call these ephemeral wetlands. And I got to thinking, okay, maybe there's a question here that I need to address. I'll tell you what that question is. During the summertime, during the springtime, okay, we go out there and we plant our seeds, most of them being treated. In the springtime, we get an abundance of rainfall, okay, when those neonicotinoids are still localized and in their very highest concentrations. And we see scenes like this quite a bit, okay? And in fact, a lot of farm fields have ancestral wetlands that are year after year planted through. And in a lot of those years, they never produce a crop, right? They just contain water. They're sort of these wetland pools that just persist. And they attract an abundance of wildlife. Okay, so you can see here's one of my field sites. Hopefully you can hear this. No birds out there in this one. I scared them all away, but it's just a cacophony of amphibian noise occurring at this embedded ephemeral wetland. Okay, why are wildlife attracted to these places? Okay, first and foremost, I thought we'd have the ornithologist in the house, and he'd be able to tell us all about why birds like these places. But I'll just pretend to be an ornithologist and tell you why birds like these places. They're wide open, okay? When it comes to being predator vigilant. It's nice to be out in the middle of a field and you can see a coyote coming from a mile away. Okay, so that's first and foremost pretty attractive. They're warm and they're open. Okay, we're starting to again to get into that time of year where we can actually have most natural bodies of water still iced over, but these pools in fields are wide open. So it's really super attractive to waterfowl. And they're definitely not devoid of resources. If you walk out into these spaces, they're just littered with dead worms. Okay, there's seeds of both this year's planted crops and last year's seeds that were unharvested. And these soils, these waters are oftentimes full of minerals. And we get back to a question that was asked earlier about honeybees, and this is an idea that I would really like to follow through with in a subsequent research study. Insects, pollinators, butterflies, bees, if you were to give them a right off the tap bowl of water, and if you were to go out and grab some mucky water from your driveway, they will flock to that mucky pool of water. It's called puddling. This is why, if you see, why would that butterfly be sucking up that nasty looking pothole on the road? It's because a lot of these environments are pretty scarce in minerals, and they will sap up this stuff like it's Gatorade. Okay, so that's another reason why some of these ephemeral ag wetlands are really attractive to things like honeybees in the springtime that are coming to hydrate themselves at open water pools. Okay. My main research question after looking at these things is, I wonder, okay, after we plant through these areas and we get springtime rainfall, how much of that water-soluble insecticide is present in those ephemeral pools for how long? Okay, so what I did is I went out to 20 soybean sites in Meeker County, Minnesota, south central Minnesota. 18 of them were treated with the same exact imidacloprid concentration. 2 were organic fields. After a rainfall event, I'd wait for 24 hours, and then I'd go scope out which of my ephemeral pools were still containing water. And I would collect water from them using these disposable pipettes out in the field. Okay, to start to get an idea of the wildlife usage of these ponds, we set out some sound recorders. And then at the end of the season, I went into the lab and quantified the neonics in these wetlands. Okay, this is no real surprise to you since I already showed you a video of a pile of amphibians singing in these fields. But the audio moth recorders, these sound recorders, captured an amphibian community that is actively utilizing these in-field wetlands for reproductive activities. I'm still going through the recordings to parse out all the bird sounds that are in them, but it's a lot. I need someone's help to do that. So that's going to take me a little while. Now, I guess for the kicker, like in my mind, the recipe was right for this water to contain pesticide, but you never know how much, you never know for sure if what you're collecting is going to produce anything. So creating a graph like this for a person like me is really exciting, because all of a sudden you're like, I did it. Okay, it's there. I found it. So here we have on this graph, the bottom is early in the season, May, and then I was able to collect after 6 rainfall events throughout the summer. Okay. And over here on the left-hand side, you can see the concentration of the insecticidal seed treatment there. So early on the season, after only a few of the soybean fields had been planted, we had this big rainfall event. And before the seeds were ever germinated, I was able to go out and collect some water. And that is by far the highest concentration that I found. Okay, 27 parts per billion. And I'll try and put that into context here in just a minute. And then we see throughout the growing season, like we saw in the rest of the cases where we quantified neonics in the environment, they dilute, right? And they metabolize in the environment. But look down here at the graph, at this table, you get a better idea of how long the neonics stay around. But what do those numbers really mean here? We look again back to our guinea pig, the honeybee, and look at the LD50 values. You look throughout the literature, a 48-hour exposure to a water source at 0.6 parts per billion, a 48-hour exposure where that is their water option, 0.6 parts per billion is enough to kill half of the honeybee population fed. Okay, so 0.6 is still present in the water through the beginning of July in these cases. A single feeding enough to kill half of the honeybee population is 38 parts per billion. You see that's about right here. So we have at least one wetland early on in the springtime where any bee that visits that particular wetland is done. Okay, is Dunsky. Okay, what else? Let's talk about more than just honeybees here. If you look at aquatic organisms, here we've got mayfly nymphs. Their population killed 50%, 100% reduced mobility at 1.2 parts per billion. Okay. So hopefully this isn't too data-y for people. I love this because I can understand now how much this is just a whole nother route of exposure that before this study was never talked about, was never elucidated before. This lab is quantifying insecticides in tissues of predators, okay, that lap up water in the springtime. Where is this insecticide coming from? Okay, it's hard to figure out why we might be quantifying that much pesticide in a predator. Well, okay, we start to look wherever we're looking. We're finding neonicotinoids in unintended places. Okay. What needs to happen now is to utilize this data and try and figure out what it means for the organisms that are potentially exposed to that level of a neonicotinoid concentration. I want to tell you one last very short story about one of the coolest studies that I've ever read. We've got to, in our mind's eye, travel to the other side of the world. Okay, we're going to Lake Shinji, Japan. Okay, and Lake Shinji, you can see in this image here. Normally, for my students, I'd bring up the Google map. I'm not going to do that now. You see on the western edge of Lake Shinji, you've got all these little tributaries coming in. What those are irrigation canals that are dumping into Lake Shinji from a river valley that is utilized almost solely for rice production. Okay, if we look at the lake is a very important resource. Economically, there are 3 fish that supply a huge amount of calories and support a very important part of the local economy. Smelt, eel, and ice fish come out of Lake Shinji. Okay. Smelt and eel are the bigger of the species, and they consume zooplankton. These are these bug-like critters that float around in water. The ice fish is the smaller of the 3 fish and will consume the bugs in the water, the zooplankton. They also survive on the little floating tiny microscopic plants in the water. So they're also herbivores. So they've got a different component of their diet than the first 2. Okay. In this region, in this province, they call it a prefecture, they keep incredible data, which is why this study was able to be accomplished. This is a story about the power of data, right? In 1993, the first neonicotinoid seed treatments, imidacloprid, started to be used in rice in the watershed of Lake Shinji. The watershed, again, is the rice production region that flows into Lake Shinji. The local fisheries department in the prefecture every year takes a survey of the little bug community in the water, the zooplankton community. Okay, 1993. You can see without me describing it what happens to the little bug community in the water. All right. The same department also tracks how many fish, or what poundage of fish are caught out of this resource because it is such an important economic resource for the region. 1993, when we start utilizing neonicotinoid seed treatments on rice in the watershed, when we see a decimation of the zooplankton community, our two zooplanktonivorous fish, how you like that for a term, the smelt and the eel, their populations react very, very quickly to that loss of a food resource. The only fish of the three that depends less on zooplankton and can continue to sustain their population utilizing vegetative, diatoms and algae, their population continues to persist at about the same level as it did before. Okay, and there are other examples from around the world that are starting to collect the data necessary to answer these same really hard questions to answer. How does something as simple as a seed treatment on a crop seed in an agricultural field impact a fish community down the trophic pyramid, down the food web? Okay. This paper is also just a fantastic piece of work. Please read it. It talks about the same issue occurring in these folks from the Netherlands, accumulating insect abundance data, figuring out when neonicotinoid pesticides were being used, and then correlating that information with insectivorous bird populations. All right. So, what did we talk about today? Let's review real quickly. I hope it wasn't too much of a snoozer. I know it was a long talk. I know we got into a nitty-gritty, but I think we covered a lot. Okay? And I loved it. Even if only half of the people, even if a quarter of people did, I loved it. Okay? So, what I hope we learned is primarily about what this thing is, what this weird nebulous term neonicotinoid means. It's not magic dust, right? It has real physiological consequences in an organism's body. Okay, it interferes with normal nerve functioning. I hope you know how it moves from point A to point B. I hope you now know what dictates how long it persists in an environment, why it breaks down, how it breaks down, how certain organisms may encounter it. I hope you now understand the intended use of this insecticidal class. And I hope you learned about the mode of dispersal for neonicotinoids, seed treatments, how they work. And then how finally wildlife can potentially be exposed. And ultimately, I hope that maybe some people in here were stimulated, and started thinking about their own potential work that they want to consider doing. I want to figure out, I hope this went through someone's mind. I want to figure out other community and ecological consequences of agricultural toxins. It's an awesome field of work. Okay, that's it. I'm done. We're all roasted. I can see it. Our brain cells are full, and our carbohydrates are depleted. So that's it. Thank you. And I will take any questions at this time from the room, and I'll repeat them if they're from the room. And Ryan, if we've got anything online, please have at it.
So we have some good stuff from online to kind of kick us off here. Thank you, Mike. That was, yeah, I don't think anybody online thought it was boring. We have some good comments. I'll start off here. One is bumblebees and other birds and insects drink the guttation liquid at the tips of leaves. I was wondering, and that reminded me of extrafloral nectaries in some of the plants. Would you mind touching on what extrafloral nectaries are and how that can expose insects to these pesticides?
Yes. What is an extrafloral nectary? Okay, so let's take sunflowers for example. What is it? Let's answer a question. What is a nectary? A nectary is a gland on a plant or an organ that exudes sugar water. Why would a plant do that? Well, it's to attract something else. It's not helping. It's not bathing itself in sugar because it needs a good sugar scrub, right? It's exuding sugar to attract pollinators, usually to a floral resource in a flower, right? That's why it's sugary water in the bottom of a flower. There's actually a ton of plants that have something called an extra floral nectary, which means that there is a nectary doing the same thing, exuding sugar water that's not associated with a flower. Sunflowers do this right here. If I'm a sunflower plant and these are my leaves on my sides, right here is my petiole connecting my leaf blade to the central shaft of the sunflower. Right here at the junction between the leaf and the petiole, there are extrafloral nectaries, little glands that push out sugar. And they do that all season long. And what they're doing is they're paying it forward. They're giving this expensive nutrition to ants, to wasps. Spiders drink it, to lady beetles. What they're doing is they're saying, hey, I'm here to pay and attract my army of predatory insects to be here in full force for when I eventually open up my flower and I start the real dangerous business of creating structures to put my genes in, seeds, to disperse them and create the next generation. Okay, so that's what an extrafloral nectary is. Legumes, so beans and peas, a lot of them have it. You know, other very common in dicots will have these things. And the question probably is alluding to, is there a neonicotinoid or a systemic pesticide that's found in extrafloral nectar? Yes, that was actually part of my initial sunflower project that I did not present here today, but that was part of the deal. In the greenhouse, I grew in a super hot greenhouse, increased the humidity, and we were out there on these little nectaries scraping off nectar and sugar from those nectaries, and then I quantified them. And yes, we found neonicotinoids present in those nectaries. The other aspect you talk about was guttation fluid. Okay. I get excited about this stuff. Yeah, I hope you guys are interested in this. Guttation fluid. Who's heard of guttation fluid? Okay. 2 people with PhDs in the room have heard of guttation fluid. What guttation fluid is in a growing leaf meristem, at the very growing tip of a leaf, you have cell differentiation happening like crazy. As part of that crazy cell differentiation, sometimes in an actively very fast growing, we'll use corn or a grass tip, they grow so fast, there'll be a direct connection from the roots of a plant through the xylem or the water tubes all the way to the tippy top of that growing leaf, and there won't yet be the structures to enclose fully that xylem. So actually water from the roots goes straight up through the plant and actually gets squeezed out the growing tip of that growing meristem. And that's what guttation fluid is. And very interestingly, in a very young plant, let's say you've got a corn plant that's just a few days old and it's a hot day, corn grows fast. And right away in the morning, there's tons of pressure down in those roots. The plant is drawing in water like crazy, and it's actually pushing water that is of the same nutrient concentration as right around its root system, pushing it right out the top of that leaf. And whatever is in that soil water is also present in that droplet of guttation fluid. So what's dissolved in the soil water right around a treated corn seed, soybean seed, canola seed, whatever plant, it's the neonicotinoids. So yes, that work has been done. You can find a pile of neonics in guttation fluid. And that was a really long answer for a pretty short question.
No, that's really good because most people, I don't think, realize that there's those potential for exposure. Thank you, Mike. We have another good one here. Why do you think the metabolites increased by a factor of 10, between the thimethoxin you presented and then the clothianidin?
I don't know the answer to that. Besides, it's commonly known that some of the neonicotinoids very quickly and readily are converted to another neonicotinoid form in a plant. It happens pretty fast. So the fact that we found the majority of toxic material being the converted form doesn't surprise me too much. That's not too much of a surprise based on other evidence. But I can't tell you how physiologically in the plant it's converted from one form to the other. But yeah, I'll leave it there. It's some neonicotinoids. See, that's why when you're trying to look for thiamethoxam, when that is your seed treatment of choice, it's widely known now that it really quickly turns into clothianidin in a plant. It doesn't happen when you use imidacloprid, the most common neonicotinoid seed treatment. That hangs around as imidacloprid for longer. So why certain neonicotinoids are changed within the plants, I don't know the answer to that.
We have some questions about research on neonics. I'm going to combine a few here. One is, why isn't this type of research done prior to the release of these chemicals and addressed prior to them becoming available on the marketplace? And do you know of any studies where they are examining the presence of neonics in human organs?
In humans, human tissues?
Yep.
Yeah, no, I don't know. No to that one. There hasn't been, to my knowledge, any sort of, well, no, that's not true. I know that in certain tissues, like urine, for example, neonicotinoids have been found in human urine samples. But there has not been, I guess for pretty obvious reasons, a sort of comprehensive trial of direct effects of neonicotinoids on people. When somebody asks me that question, like how toxic is it to humans? That's a good question because we haven't done a whole lot of research on that. What I can say is that I have read some really grisly examples of human beings utilizing neonicotinoids to end their own lives. So those examples and those clinical reports do exist. So that's maybe where I'll leave that one. What was the first? Okay.
Why isn't this research done before the release of these chemicals?
Yeah, well, unfortunately, a lot of that research is, it's done on a very curtailed number of organisms. And the comprehensive analysis of what a novel compound might do on an ecosystem scale to affect entire food webs
I mean, that kind of research it's not easy to do, which is probably the reason why it's not done. It just isn't done on that scale to take into effect, to take into account what might happen on this food web scale. It's just not done. You know, when something is approved, it goes through a certain number of, let's call them guinea pig organisms, and if they pass through enough of those guinea pig organisms, and they don't directly affect their population bad enough, well, they can then move on to the next step of approval. It's really unfortunate, but it's not the first example of something where we, approve and find that the worst of the effects of something emerges later on, and a lot of the effects are unintended, unexpected consequences. So it's really, I guess it's kind of par for the course, really.
Yeah. We have a few more online, unless there's some coming in from the room there.
Bobby?
Did that have any effect? And was there, is there any follow-up on that? I mean, I think also
Yeah, that I know that research for the
Could you
follow-up.
repeat the question?
Yes. So there have been places around the world where there was at least a period of, or a full-on neonicotinoid ban, and the room is wondering if that ban has had a positive effect on communities that were suffering pre-ban. And that work is being done right now. I know that those studies are coming out. And John, have you read, I know that there's been really recent stuff published out of Europe that have been looking at, has the ban worked or not? I haven't read it yet.
It's starting to roll out now. They're looking at bird populations specifically and showing a lot of that. But
the birds
really depends on who you talk to.
Right. I know the bird work that's just come out, and this is very, very fresh. And I think at best I can say the data is messy right now. But the researchers, anyways, that published the most recent study, they said it looks like there's maybe a rebound occurring in some of the birds. This is hard. It's difficult to attribute one aspect of a community change to what's occurring across an entire food web. So it's tough, but I think it's going to, I don't know, hopefully we'll gain more clarity. We will gain more clarity over time. And I can't tell you how long that's going to take. Any more room questions? None from the room, Ryan? Any questions?
We have some from online here. A few here. They ask, in your study, the original product converts to a more potent form in the cover crops. What happens when the cover crops break down? Is the cycle over, or does the more potent form survive in the soil?
Yeah, so we look back. If we think back to those slides, maybe I can just scroll up to it. It wasn't that long ago. So as is the case with any neonicotinoid study that you look at, just like that one, you see this decay of the neonicotinoids over time. It's taking me a lot longer to get back there. You see this decay over time, okay? And this is so dependent on location and environmental conditions on any given year. I've read studies where corn was grown in a situation where the corn came up, it grew to about a foot tall, and then it just baked and just died. And that corn, the neonics in that situation persisted in that corn tissue for a long time. It persisted at a very high level relative to corn plants that get a lot of precipitation and they grow, and just naturally the compound's able to dilute within the plant. Within the plant, it's being degraded. That moisture in the soil increases microbial respiration, and they're looking for things to decompose, and this nitrogenous compound, these neonicotinoids, are just degraded. So how long a neonicotinoid does persist in the environment has everything to do with microbial decomposition rates. So how friendly your soils are, how much organic matter there is, what kind of temperature and moisture conditions there are, has everything to do with how long a neonicotinoid takes to break down. But it's just knowing those components, you can kind of then conceptualize what scenarios leads to the long time persistence of a neonicotinoid versus a situation where they degrade to the point where they are undetectable. Okay, it has everything to do with, if your environment is more suited to microbial activity, they're going to break down faster. You're going to not see them or be able to quantify them a lot sooner than if you have a heavy clay soil in a super dry environment and they just sit there sort of glommed on like magnets to clay in the soil, and there's nothing that's there breaking them down. I hope that is enough to answer that question. Question in the room.
Should farmers use neonicotinoids?
It's a good question. Like tying it up with a bow. The question is,
repeat the question, please.
Yeah, should farmers use neonicotinoid pesticides? And what I can tell you is that, well, let's just talk pure economics at the moment. My very last slide, I even snuck in a final slide at the very end of this entire thing. I'll keep scrolling as I talk. It is very, very rare where a study concludes that a neonicotinoid pesticide is in any way, shape, or form positive in terms of economics. Okay. So that's kind of perhaps a weak answer. Economically, the vast majority of situations don't call for neonicotinoid use, because when they are used, and the studies have been conducted, they're on the screen right now, very big comprehensive studies, they don't show an economic benefit. Okay, and that is for whatever reason. Usually, okay, I'll take the study on the left. This is a massive study that they looked at. Okay, almost everybody uses neonics on their soybeans, at least in the upper Midwest, right? And the idea is that aphids are this fluid feeding insect, and neonics should be able to take care of them. This study found that no, in almost zero cases did the neonicotinoid seed treatment in soybeans ever prevent a field from being sprayed later on in the season for soybean aphids. So it's a redundancy. In the vast majority of the cases, for whatever reason, the neonicotinoid insecticide is just at a low enough concentration, has been metabolized, dispersed enough to just not really affect soybeans, which is what people say it's used for so often here in the upper Midwest. Okay. Should they be using them is a whole nother question. So economically, no, it's really, in the vast majority of situations, probably a bad idea. I will acquiesce and say, if your system is degraded to the point where you have, and you're growing a crop that there is a substantial pest of, say, the seed, the crop seed that you put in the ground, and there's a pile of crop seed feeding insects, that's probably where you do see an economic return. But again, that's in a situation where you've built up this population of herbivorous insects through a very simplified agricultural regime to make them kind of a substantial pest. And it's a last ditch effort. It's kind of the only thing that saves you in that situation. But please, in the vast majority of situations, try without it, because it is the first thing when I talk to farmers that are trying to utilize an ecological based pest management regime, right? Utilizing the complexities and stop blocks that nature provides for free. The first thing that they tell me that they get away with without any sort of negative consequence is planting naked seeds. They get away with it. It's just they're just not that, they're just not as effective as they're just not that effective to be economically viable. I hope that was somewhat of an answer.
The answer.
That was great, Mike. That actually took care of a good question we had online that was very closely tied to it. Maybe one last thing. We want to respect everybody's time, and you've been speaking for an hour and a half for us. One last question here. This one kind of has to deal with the application of neonics outside of agricultural settings. I know it's been in the press, and this person indicates, there's been a lot of like cities and in nurseries and landscaping. They use neonics quite a bit.
Yeah.
They're hoping if you could discuss the potential effects of those in the flowering ornamental landscape trees and the effects on biodiversity. Can you share your thoughts on this subject or any research you've come across?
Yeah. Unfortunately, you're exactly right. In a lot of already pre-potted ornamental plants, a lot of those seeds, and I can't speak for all of them, but a lot of them do start their life as a neonicotinoid treated seed. And you also do need to be aware of, when you're starting your garden, that yes, a lot of garden seeds are also seed treated. And they have the potential to have the same exact effects as they do under normal agricultural situations. Okay. They will most likely, if they're still present in high enough concentrations, be found in the pollen and nectar of your ornamental flowers. They will certainly be in the tissues of beans, peas, seeds, squash in the garden. Okay, it's the same concepts apply as what we were talking about in this more agrarian topic that we got done with here. So what I would say is that, and I've started to do this over the last few years, you can ask your greenhouse, and a lot of them are starting to become aware of how important this issue is to their customers. And they will tell you, well, if they don't know, then there are greenhouses around that do know, or do intentionally start their vegetable starts, start their ornamental starts with completely untreated naked seeds. So if you are at all concerned about it, if they're not sure whether they do or not, best practice is to find someone that does know. And this is not like, they're not going to look at you like you're crazy. Like, there are plenty of greenhouses and nurseries now that are aware of this aspect of horticulture and smallholder gardening, and they will intentionally use untreated seeds.
Wonderful. Well, I think we should end it there and thank Dr. Bredeson for all this good knowledge he shared with us. For the folks online, thank you for the great questions. And remember, this talk was recorded and it will be posted to our YouTube channel. So if you know of anybody that couldn't make it today, please let them know. You can find it on our YouTube channel here within the next month or so.
Hey, Ryan.
Yes.
One last interruption here. I was just handed a card.
Okay.
It says, please sign up for the upcoming Ecdysis webinars at ecdysis.bio/events. So if you want to be on sort of the in the know list for upcoming events, that's where you need to go, ecdysis.bio/events, or probably get onto the Facebook page.
Yes, wonderful. Thank you for that. Yep. And next month on March 20th, we'll have Dr. Judy Wu Smart discussing the next phase in this neonics series, the effects of neonics on honeybees specifically, and a lot of the neat research she's done down at University of Nebraska. So please join us then and look for those webinar announcements where Mike just described. I guess that's all we have, and just thank Dr. Bredeson again for sharing, and thank you for the audience for showing up, guys, and good questions.
Thanks so much, Ryan. Appreciate it.